Good morning. I'm Dr. Natarajan Suresh, Professor of Pathology, Sree Balaji Medical College and Hospital, Krompet, Chennai. It's a very interesting topic which I'm going to discuss today, which is about oncogenesis, and which it's very fascinating and though complex, has got a very wide ramifications in the field of medicine. So the competencies and what we're going to learn, the specific learning objectives are, we're going to enumerate the process of carcinogenesis and enumerate the carcinogens causing the cancer. As all of you know, the cell goes through what is called the cell cycle before it forms daughter cells. So you have a mitotic phase and what is called the interphase. So in interphase, you have three phases. You have the G1 phase, you have the synthesis phase, and the G2 phase. So there are two phases called the action phases. Usually the S phase and the M phase are called the action phases. We'll come to that in just a brief moment. So how does a cell proliferate and how does the cell move through its life cycle? Not all cells go through this cycle. There are some cells which proliferate rapidly. There are some cells which remain quiescent, and some cells don't proliferate at all. So that is cells in the skin, in hair and GIT. They turn over rapidly within a single day. So that's how any anti-cancer drugs cause hair fall or cause GI disturbances like diarrhea, while these anti-cancer drugs don't affect liver or don't affect the neurons. A person with cancer doesn't get a neurological problem, but he will get all other problems. This is because the cell turnover is high and these cells get affected by the cancer drugs. So we'll come to this process, how it happens. So as we said, there are three phases and there's one mitotic phase. So the cell growth occurs through G1 and it is driven by what are called proteins called cyclin D and CDK4. And there are three restriction points. You have a G1 restriction point, and then you have G2 checkpoint, and then you have a M checkpoint. The cell is driven through G1 to S, where there is a DNA synthesis, and then from S phase it goes to G2 phase and there's a cell growth, and then in M phase the daughter cells are produced. Sometimes not all cells have to undergo this phase. Some cells will undergo and be quiescent or will go into a resting phase, and this phase is called G0 phase. This restriction point, G1S restriction point, is a very important restriction point because before the cell DNA synthesis occurs, a mutated or aberrant DNA synthesis is corrected at this stage. So we come to the molecular basis of cancer. Non-lethal genetic damage is the basis of carcinogenesis. Genetic hypothesis of cancer implies that the tumor mass results from a clonal expansion of a single progenitor cell that incurred the genetic damage. So what do I mean by that? Non-lethal genetic damage, a normal cell gets induced by a carcinogen, and then an initiated precursor with stem cell-like properties is produced. Then there are additional driver mutations, and then this forms the founding cancer cells with additional mutations, emergence of subclones, and then genetically heterogeneous cancer is formed. So you have initiating mutations and acquisition of different hallmarks of cancer, which will come again, we'll come to it, and then finally a tumor is produced. So how does the regulation of cell cycle occur? Any external stimuli leads to stimulation of growth by growth stimulating gene, which is called proto-oncogene. This should be stopped by a gene which will control the growth called the tumor suppressor gene. And any mutated gene will be either repaired by a DNA repair gene or killed by a gene called apoptosis gene. So if I can give an example of an injury of skin when more skin cells are needed. The growth factor PDGFR alpha from the platelets induce the growth receptor to induce proto-oncogene. The proto-oncogenes moves the cell cycle forward, producing adequate skin cells. Tumor suppressor genes halts the cell cycle when the need is over. Suppose you have a wound, you don't want the skin to grow forever. The apoptosis genes checks for mutated cells and repairs them and kills them. So at the end, you get a healed skin. Unfortunately, this doesn't happen in the heart or brain as the cell cycle is nil, causing permanent neuronal damage or myocardial damage. So what is the principle of carcinogenesis? Neoplastic transformation is a progressive process involving multiple hits or genetic changes. These multiple hits can be because of radiation, can be because of chemical, can be because of viruses, or can be because of microbes. Any of them can cause a neoplastic transformation. Alteration in DNA causes changes in one or more of the following type of genes. One is proto-oncogene, others is tumor suppressor genes, then the genes which regulate apoptosis and DNA repair genes. So what are the molecular hallmarks of cancer? You have multiple hallmarks of cancer. One is it evades growth suppressors. It promotes replicative immortality. It has tumor promoting inflammation. You have activating invasion and metastasis. You have genomic instability. It resists cell death. It induces angiogenesis. It deregulates cellular energetics and sustains proliferative signaling with immune destruction avoidance. So now let us come to one of the most important concept, which is the most important takeaway from this discussion. So what is the difference between a proto-oncogene and an oncogene? If we just take an analogy, proto-oncogenes are like the accelerator of a car moving the car forward. If a car has got an accelerator, proto-oncogene is the accelerator, and it is a normal gene of a normal cell. A normal cell requires proto-oncogene for its normal cellular functions. Now in case the accelerator gets blocked or it gets stuck and the car keeps on moving, the car has got no way of stopping. Now this is what is called, now proto-oncogene has transformed into an oncogene. Now a car is moving, the accelerator is being pressed and it has become an oncogene. How can the car be stopped? The car can be stopped by only one way, and that is by a different set of genes, which we shall come to know. We'll come to it a little later. So now we got an analogy. Proto-oncogenes are normal genes involved in cell growth and function. Oncogenes are mutated proto-oncogenes. There is gain of function mutation that will cause uncontrolled cell proliferation. So this is the mechanism by which the cancer signaling pathway occurs. You have a growth factor which gets attached to a growth factor receptor, which are tyrosine kinase usually. And now this will activate the RAS protein into an active RAS. Now this active RAS has got a down signaling pathway. It activates RAF, it activates MAPK and PI3K. There are different proteins which are activated, and these activate transcription. Now you need a growth factor and you need a growth factor receptor in normal conditions. But what happens in a tumor? This normal signal becomes autonomous. There is no more a requirement of a growth factor. Without a growth factor attaching itself to a growth factor receptor, the tumor can start producing its own signals, and there is a autonomous signaling pathway which gets accelerated with each cell growth. So there is no more a negative checkpoint, that is that you need a growth factor or you need a growth factor receptor. So you have different factors like PDGFR, TGF alpha, you have tyrosine kinase receptors, you have KRAS, NRAS, HRAS and RAF. Mutations can cause melanomas. Now we'll come to what is called tumor suppressor. Now you have what is the second component, how a tumor is produced is because of insensitivity to growth inhibitors. These are because of tumor suppressor genes. As we said in proto-oncogenes, if they can be likened to an accelerator, tumor suppressor genes can be likened to a brakes in a car, which apply a brake to a cell proliferation, and abnormalities in these genes can lead to a failure of growth inhibition. So what are these tumor suppressor genes? These are the retinoblastoma gene. This was the first tumor suppressor gene which was found in a patient with retinoblastoma. It is the tumor of the retina in small children. And second is P53 gene, which is is the guardian of the genome or the molecular policeman of the cell. And then you have adenomatous polyposis gene. The loss of function mutations leads to cancer. In proto-oncogene, it was gain of function. In tumor suppressor genes, it's a loss of function. So what is retinoblastoma gene? This is also called the governor of cell cycle. So you have what is called the Knutson's two-hit hypothesis. That is, two alleles need to be mutated in retinoblastoma. In familiar cases, the children inherit one defective copy of the retinoblastoma, that is the first hit, and one normal copy of retinoblastoma in the germline. Retinoblastoma develops when the normal RB allele is mutated in retinoblasts as a result of spontaneous somatic mutation, which is the second hit. Because second hit seems to be virtually inevitable in a small fraction of retinoblasts, most individuals inheriting a germline defect in one RB allele develop unilateral or bilateral retinoblastoma. So what is P53, or it's also called the policeman of the cell cycle or the guardian of the genome? Once activated, P53 thwarts the neoplastic transformation by causing a transient cell cycle arrest. It induces senescence, the cells permanently are arrested, and it also induces apoptosis by transcripts called BAX and PUMA to kill the cell. With loss of P53 function, DNA damage goes unrepaired, driving mutations accumulate in the oncogenes and other cancer genes, and the cell marches along a dangerous path leading to malignant transformation. APC is the gatekeeper of colonic neoplasias. These tumor suppressor genes function by down-regulating the growth-promoting signaling pathways. APC is a component of the WNT signaling pathway, which has a major role in controlling cellular growth and differentiation during embryonic development. So what is the Warburg effect? Warburg effect is an important effect by which the tumors continue to grow even though there is only aerobic glycolysis. Aerobic glycolysis, the tumor cells eat more glucose than normal cells. This activity is seen in PET scans via 18F fluorodeoxyglucose injection. Why do cancer cells go for glycolysis? Only when if only 2 ATPs are produced instead of oxidative phosphorylation in which 36 ATPs are produced. The reason why a cancer cell goes for a glycolysis where though it's getting less energy instead of oxidative phosphorylation is because aerobic glycolysis provides rapidly dividing tumor cells with metabolic intermediates that are needed for synthesis of cellular components, whereas mitochondrial oxidative phosphorylation does not. How does the cell then evade cell death? So cell death is evaded. Tumor cells frequently contain mutations in genes that result in resistance to apoptotic cell death. There is loss of p53 function and there is an overexpression of BCL2 family. No apoptosis is seen in B-cell lymphoma. The cell needs to grow and for that it requires blood supply. Even if a solid tumor possesses all the genetic aberrations that are required for a malignant transformation, it cannot enlarge beyond 1 to 2 millimeter in diameter unless it has a capacity to induce angiogenesis. Lack of oxygen in tumor causes H1F alpha, 1 alpha to produce VEGF to produce new blood vessels. The loss of p53 does not stop VEGF, so more blood vessels are produced. VEGF is also called vascular endothelial growth factor. And then the tumor cells also evade the immune surveillance by multiple factors. Normally, the mutated normal self proteins there is no normal host cell displaying multiple MHC associated self antigen, so there is no T cell response. But if the tumor cells express different types of tumor antigens, the protein variants are created by different driver or passenger mutations. So then there is the other ways how carcinogenesis can be produced is by chemical carcinogenesis. You have different chemicals, the long list of chemicals, direct acting carcinogens, procarcinogens, which can all cause metabolic activation and produce cancer. All initiating chemical carcinogens are highly reactive electrophiles that have electron deficient atoms that can react with nucleophilic electron rich atoms in the cell. Their targets are DNA, RNA and proteins. So direct acting carcinogens do not require metabolic conversion to become carcinogenic. Example, alkylating agents. Indirect acting carcinogens require metabolic conversion to become active carcinogens. The carcinogenic products are called ultimate carcinogens. The most potent indirect chemical carcinogens are present in fossil fuels. Benzopyrene are formed during the high temperature combustion of tobacco and cigarettes and are implicated in the causation of lung cancers. Polycyclic hydrocarbons are produced from animal fats during the process of broiling or grilling of meats and are present in smoked meats and fish. These are metabolized by cytochrome P450 dependent monooxygenases, and the genes that encode them are polymorphic and differ among individuals. So what are the molecular targets of chemical carcinogens? The aflatoxins from Aspergillus causes hepatocellular cancer in a person with mutations in TP53. So there is AG and CT transversion, GC to TA transversion in codon 249 that produces an arginine to serine substitution in the p53 protein. The other carcinogenesis is because of ultraviolet rays. The radiant energy in the form of UV rays of sunlight or as ionizing electromagnetic and particulate radiation is mutagenic and carcinogenic. The exposure to UV rays derived from the sun, particularly in the fair-skinned individuals, is associated with an increased incidence of squamous cell carcinoma, basal cell carcinoma and melanoma of the skin. The UVB light is carcinogenic because of its ability to cause pyrimidine dimers to form in DNA. Then what is microbial carcinogenesis? Even microbes can cause cancers. The most important is oncogenic RNA virus caused by human T-cell leukemia virus type 1, oncogenic DNA virus, HPV virus, Epstein-Barr virus, hepatitis B and C virus, and bacteria causing cancer, which is Helicobacter pylori. What are the oncogenic RNA virus? Oncogenic RNA virus are human T-cell leukemia virus type 1, which causes adult T-cell leukemia/lymphoma. This virus targets CD4 cells. The viral RNA contains TAX and HBZ genes. The TAX is needed for viral replication because it stimulates transcription of viral RNA from the 5' long terminal repeat. HBZ is a transcription factor. TAX and HBZ alter the transcription of host cell genes and interact with certain host cell signaling proteins. We come to the end of this talk with a small discussion on the oncogenic DNA virus. The oncogenic DNA virus are mainly human papilloma virus, which is one of the most important virus, which causes cervical carcinomas. There are two types. They are low risk, which are HPV types are 6, 11, 42, 43 and 44, and these cause genital warts or benign lesions, and they don't cause cervical cancer. High risk are 16, 18, 31, 33, 35, 39, 45, 51, and these types are isolated in cervical cancers. And last but not the least is the Epstein-Barr virus, which is linked with the Burkitt lymphoma. LMP1 is the gene in the virus, which is an oncogene causing B-cell lymphoma. LMP1 activates the NFK beta and JAK-STAT signaling pathways and promotes B-cell survival and proliferation. Another gene is EBNA2, which activates cyclin D and SRC family of proto-oncogenes. So this is present in Burkitt lymphoma, which is very common in Africa. So today we discussed about the hallmark and the entire process, how cancer is produced, what are the mechanisms, what are the pathways, and the different carcinogens which cause cancer. Thank you. Let me just recapitulate what we studied today. We discussed about cell cycle, molecular pathways of cancer, hallmarks of cancer, what are proto-oncogenes, what are oncogenes, what are tumor suppressor genes, how does tumor evade apoptosis, and how does it evade immune surveillance, and different carcinogens like radiation carcinogens, chemical carcinogens, and microbial carcinogens. Thank you.